Hi, my name is Aaron Keaton, and I'm a senior in the Environmental Studies Department at Keene State College. My project is identifying local mushroom species in and near Keene, New Hampshire to develop a local field guide. My faculty mentor for this project is Dr. Nora Travis. To begin, the reason why I wanted to do this project is because mushrooms are an incredible species. I wanted to learn more about them and how they interact in our local environment. So mushrooms are the fruiting body of mycelium. Mycelium is the thread-like structure beneath our soil that has long been known to create fundamental symbiotic relationships between fungi and other organisms such as plants and trees. Mycelium helps transport nutrients between these organisms and also helps regulate carbon and nitrogen ratios in the soil. Along with transporting nutrients, they are also known to be overall communicators between organisms in the environment. Mushrooms are decomposers, breaking down dead organic matter and putting that nutrients back into the soil to be used again. So mushrooms are recyclers that bridge this gap between life and death and keep everything going in the environment again and again. Mycelium and mushrooms together are incredibly important to the health and structure of forest ecosystems. Out of the estimated 1.5 million species of fungi that exist, only 5% have been described in the literature. This makes the species severely under-researched and the need for local field guides incredibly important. So for my project, I wanted to find locally found mushrooms in the Monadnock region and describe them so that anyone else could go out and learn more about them. I believe that if we learn more about this species that is essential to the health and structure of our ecosystems, we will be better able to preserve them. So for my project, I went to some local forests and recreational areas around Keene, New Hampshire. This included Goose Pond, the forest at Surrey Dam, Fitzwilliam Depot area, and Chesterfield Gorge. I spent approximately 40 hours on field analysis this semester and found approximately 20 or more fungi species. This included going to all the sites listed in figure 1.1, taking pictures, and writing any detailed notes that I felt were necessary, as long as taking GPS coordinates of each specimen. So for this semester, I basically spent all my time going out and learning about mushrooms in our area and just finding what I could find, taking all the pictures and working on identifying them. So to identify mushrooms, you're going to be looking at visual characteristics. If we look at figure 1.2, that's an anatomy of a traditional mushroom most people would think of, cap and stem. So you're mostly going to be looking at the cap, what type of scales, if it has scales or warts, if it has gills, pores, or spines. Even though, like I said, this is a traditional mushroom, they come in all shapes and sizes. So if you look at figure 1.4, Serena unicolor, this is a mossy polypore. There's a more thin chip-like structure, opposed to figure 1.5, Phlemulina velectipes. This is a traditional mushroom, like I said, cap and stem. And then figure 1.7, Tremella mesenterica, the witch's butter. This is a jelly-like fungi. And just to note, all these mushrooms in the preliminary results are mushrooms found on either trees or dead decaying matter. I didn't mean to do this. I found a lot of mushrooms that grow from the soil, and the only difference is mushrooms that you find sprouted out of the soil are a part of a larger mycelium network, while ones found on dead decaying trees are just there to get the nutrients from that tree for the time being. So like I said, you're going to be looking at visual characteristics to identify a mushroom, but something else you can do is make a spore print. So figure 1.3 is a picture of a spore print. You would make a spore print by taking the cap of a mushroom, cutting it off, laying it flat down on a piece of paper. You would then drop some drops of water on it to promote the release of spores, and then you would cover it with a cup, and a few hours later you would find a spore print. They come in different colors and shapes, and this is what you can analyze further to identify the mushroom. So for next semester, I'm going to be working on taking all the pictures and information I gathered this semester, identifying the mushrooms, making spore prints to make a field guide that anyone can use, local hikers, nature lovers, or schools, to learn more about the mushrooms in our area. I would like to thank Dr. Nora Travis for guidance and support through this whole process, and thank you to Dr. Cynthia Hayes in Bio for allowing me to use microscopes. Thank you again.